All right, now that we've mastered all of the organelles inside of the cell, it's time to look at what initially appears to be probably the simplest of the structures, seeing how it's only 2 molecules thick. The cell membrane on the outside of the cell, or on plant cells just inside of the cell wall. And again, it appears pretty simple, but it has probably one of the most important jobs in the entire cell. And you recall that that is simply the function of, or one of its major functions, to regulate what enters and exits the cell. And we want to just look at the composition of a cell membrane and how exactly it is able to do that. So remember, as we said, it's regulates what enters and exits the cell. A lot of people say it controls or it decides. We want to be careful. It doesn't decide. It's just a molecule or a large complex of molecules, of lipids. Remember what lipids are. And it's really just an interaction of the molecules, how they behave around the membrane, and whether or not they have certain chemical properties. And we'll look at that in detail. And it also does provide a little bit of support and protection. The protection really being that it regulates what enters and exits the cell. But also think about it does hold everything together. The cytoplasm and all of its organelles are contained inside of the cell membrane. So as we said, it's a thin bilayer. Now, the term layer, obviously, we know that that is a, well, define the word with a word, a layer, but bi meaning two. So literally, it's just a two-layered structure made up of lipids and proteins. Now, if we zoom in, this looks possibly initially a little bit complicated. Oh my gosh, Bentley, what is that thing that you have there? And this is very simple. This is our cell membrane. This out here would be outside the cell membrane. This would be inside. And if you look, each one of these, this little balloon guy with 2 legs, that is a lipid molecule. So you can see we form a vast sheet of these guys stuck together. And on the lower end, there's another layer down there. So you have a bilayer, a layer of lipids and a layer of lipids. Now remember, properties here, this outside of the cell, these regions here, these lipids, these phosphate groups are polar. They're going to be attracted to water. They're going to love water. Inside here are nonpolar. They're the opposite. They're the fatty acid chains, and then again the lower layer is polar down here. So we have a weird protective layer that is attracted to water out here, hates water and charged particles here, and loves water on the inside. So it actually is also fairly complex. That's pretty simple, but the other really important part that we're going to focus on are right here, these light blue guys, and those are, as we can see, those are proteins. They have different functions. Some of them can act, as we'll see, as gateways, allows molecules to move in and out of the cell. Well, in this case, it's moving in. Some of them can be, as we see here, receptor proteins. They have specific jobs to receive signals. Some of them even have, these are carbohydrate chains attached. That's for cell recognition, for a cell to recognize, hey, you belong here. So what we want to do is look at this, how this works. We can see the general structure here. And over in this inset box, we can actually see, we know what O2 is. It's oxygen molecules, and they're so small, they're able to move back and forth across the membrane easily. Other molecules won't be able to do that. So we want to look at now, we've seen the structure is, as we stated, how does this lipid bilayer actually work? How do molecules literally move across that membrane? Because again, here's the outside of the cell, here's the inside of the cell, and how is that going to actually work? Because the term for this is selectively permeable. It's actually able to, remember, we don't want to personify the cell membrane, but it's almost as if the membrane picks and chooses what can permeate, what can get across the cell membrane. And we want to look at how that works. But before we do that, we've got to be aware of the basic rule, I should say, of how and why any molecules move around, and that is based on the concept of diffusion. As we can see here, this is, in this image, if this orange zipper-looking structure were a membrane, these little purple molecules could be anything. Any type of, maybe they're sugars, maybe they're oxygen molecules, whatever they may be. And as long as they can get across the membrane here, what we can actually see is that molecules will want to move from where there's a higher concentration to a lower concentration. So This actually was a GIF file. Unfortunately, I don't know why that's not playing. However, if we were to let's throw in another series of molecules here by adding in let's do a bunch of X's. Let's say that right now, if we were to look on the left-hand side here, you can see I'm drawing in a lot of X molecules, whatever X might be. Oh my gosh, it's almost algebra. Maybe let's say they're sugar molecules. Right now, where is there a higher concentration of these X molecules? We want to look at the higher portion. And we can see that clearly there's a higher concentration. There's more of them, remember concentration per unit area. There's a larger number per this unit area on this side. There's a lower concentration of the X molecules on this side. So naturally, these molecules are crammed in, they're crowded. They are moving, and they'll actually collide off of one another. So when they hit, they'll bounce off the cell membrane, they'll bounce off of one another, and that will allow them to, as long as they can pass through the membrane, if they can fit, that will allow them to move across. And they will move from, as we said, higher to lower until they even out. So I mean, we could actually use my terrible example I just did, or even if you were to, a drop of blood were to fall into a beaker of water. That also, think about what would happen there. That also would be diffusion. If we had our beaker of water and we place, or one drop of blood were to fall in there, that drop of blood is made up of a lot of molecules, a very high concentration. But when that gets into the water, the individual molecules themselves that are making up that blood, if you will, that liquid, which is dissolved with a bunch of solids in that, will diffuse and will spread out until eventually we have a uniform light pinkish, reddish colored solution. They have spread from higher, initially inside that beaker it was lower, until we have an even concentration here. So that's really what we can think of. That's all that diffusion is the process of spreading out. Molecules, all molecules will tend to do that. So again, here's you can see here in this GIF file a cheesy example of the molecules are spreading out. And it's a kind of a poor animation, but you can see that they now are uniformly, relatively speaking, uniformly dispersed. They moved from higher to lower. And initially they're in a high concentration. All this space out here would be lower. They spread out until they've reached equilibrium. There's our diffusion. And one nice thing is that it doesn't require any energy. The cell doesn't have to do any work. It happens on its own, just from the thermal kinetic energy of the molecules themselves colliding with one another. And that's fine and dandy. So again, if we were to go back to this molecule, if this were outside the cell, inside the cell, that's an easy way for molecules to move across the membrane. What will drive them, if you will, to move across the membrane. But our problem is that, unfortunately, a lot of molecules cannot pass across the membrane. They might be too large, something like a starch. Remember, starch is very, very, very large sugars and polysaccharides. They may also be charged, actually when we say charged, what we mean is they may have, like salt, sodium chloride, is actually made up of sodium and chloride ions. Now these little charged aspects, they will not diffuse across the membrane. They'll be repelled by the membrane. And the same thing with molecules that are polar, that have very, very slight charges. These three molecules, or these three cases, they cannot move across the membrane. However, something like even a small sugar like glucose, we know our cells need glucose. They must have glucose primarily, as we learned earlier in the year, to turn into energy. So how's the glucose going to get across that cell membrane? And that appears to provide present a problem, but is actually very, very easily solved in how they move across. Because remember, we do have, in addition to the cell membrane, the lipid bilayer, we have these special doorways, proteins. And we can see here in what is actually termed facilitated diffusion. Remember, the term for that facilitated actually just means helped diffusion. We have these molecules here represented by squares, whatever they may be. They're either too large to diffuse through the membrane, or maybe they carry a charge, whatever the case may be. They can't move across the membrane on their own. However, they have these special proteins embedded in the cell membrane that act. Remember, proteins, just like enzymes, shape is very, very specific. and are attracted to each other. But that allows this molecule to pass through across the membrane. And we're still going from, again, a very high concentration out here, a low concentration in there. So they're going to diffuse. They just need to be helped by passing through the membrane. And again, we cannot forget they still are going from high to low. We haven't changed the rules. It's just the fact that they are helped across, which is a very, very nice thing. Then we can get our, let's say, for example, like we had just said, this was a glucose molecule. It has to be has to get across the membrane inside the cell. How's it going to do that? Fit by through these special proteins. That's a nice that we have these helper proteins there. And then there's even actually a third case, and this is kind of weird. This is its own unique animal in active transport. And what happens here is we have molecules actually get pumped. It's going to require energy against their concentration gradient. They're going to go from low to high. So I love, love, love this picture because we can see essentially everything that we had just talked about, all of our cases. So here is just regular normal diffusion, something like perhaps maybe this guy might be oxygen molecules, can easily diffuse across, still going from high to low. And in both of these cases here, this is facilitated diffusion. They're passing through specialized proteins, but they're still going from high to low into, and keep in mind, we keep showing them moving into the cell. It just as easily can move out of the cell, depending on the concentration gradients, high to low, low to high. But in this case here, active transport, notice that we have a low, in terms of these diamond-shaped molecules, we have a low concentration outside and a high concentration inside. Now, normally with diffusion, they would move outside of the cell if they could get across. But in this case, we're using this little ATP represents energy. The cell will actually pump them into the cell. It's going to take energy. It's going to take work. Think, picture a freshman trying to cram all of their books and bags and lunches and things inside of a locker. You open the locker, they're going to want to spill out. But here, it actually requires energy to cram them down inside of the cell, so we can build an even higher concentration. Great examples of that would be nerve cells can operate that way. Cells inside of your heart and muscles, skeletal muscles can operate this way as well, where they have to build up a concentration gradient. So this one, the only difference is it does require energy. But they're still moving across the membrane, which is kind of interesting. All right. So even one last problem here is very, very important. And that's the problem that sometimes molecules will have a tough time getting across, but water has its own special proteins, and it can move readily across the membrane in what is called osmosis. Water will still move from high to low. In this case, it's kind of difficult to see here, but these little gray dots are water molecules. We can see on the left-hand side a very high concentration of water. It will diffuse across to a low concentration. Now, if there's a low water concentration, that means there's more solute. Maybe this is salt, maybe it's water. So it almost as if water is moving to where there is, or it is this way, water is moving to where there's more solute. So as a general rule, water will always diffuse to where there is a higher concentration of solute. And in some cases, like we can see here, the water level will actually rise. And your best bet would be to think of Kool-Aid. If you make Kool-Aid or a mixed powdered drink, if you ever make that and it's too strong, it's too concentrated, if you add water to it, it dilutes it. It makes it drinkable. It actually brings the concentration of solute, now solute being sugar in this case, brings it down. So you water it down. So that's really what's happening with our osmosis in this case. So we can then, in terms of that, a couple quick terms here. Hypertonic is any solution that has a higher concentration of solute molecules. Hypotonic, a solution that has a lower concentration. And isotonic would be equal. So in this case here, if we look, the light blue being water, the dark orangish red being glucose, sugar molecules, how would you describe this side? Try to use these three terms here. Go ahead and pause it and describe the sides. So obviously you had a chance to do that. There's more sugar on this side, so this side would be hypertonic. That would make this hypotonic, and that would mean that water would diffuse across to this side to water that down. kind of an interesting scenario. So we want to end with a picture of look at what happens here when we place cells into different solutions. See if you can figure out what transpired in each case.